Hey, hello guys, welcome, vanakkam, vandanam, namaskaram. Today we are going to see genetics. Actually, class 12th, principles of variation. Okay, principles of inheritance and variation. Okay, so first lecture, I'm not going to take that much. Okay, we are going to see, but not 1 hour will be. Okay, so principles of inheritance. And variation. See, here in this lesson, okay, principles of inheritance. What is inheritance? Inheritance is the process of transformation of traits from parents to offsprings. It is a process by which traits, characteristics, chromosomes, genes are being transferred from the parents or from the ancestors to the younger generation or to the offsprings. Okay, it is the process by which the traits or the characteristics are being passed down. Okay, see, this inheritance itself is actually of two different types. We are going to read it based on two different types. That is, okay, we are going to read it based on two different types. First is going to be inheritance of one gene. Okay, then inheritance of two genes. That's it. Okay, we are going to just study the basics, the surface level. Okay, and variation. So what is variation? See, while transforming, okay, from parents to offspring, okay, genes are being transformed, right? There will be some sort of variation. Okay, there will be some sort of variation. Variation in sense, I cannot say, see, we all have some difference, okay, from our parents and to you. That means your parents and you can just take care of you two. You can just take you two into the case. You can see that there is a lot of difference. Looks will be different. For an example, you might have the eyes of your mother, okay, and you might have lips of your father, okay, but there is a variation, right? There is a variation. You will not be exactly looking like your mother, or you will not be exactly looking like your father. There will be variation. So the degree of variation, okay, the degree of change in the individuals of the same species, there will be variation. That's what it is. Okay, just in our normal English also we use, isn't it? Okay, so this is what variation we are going to study. Here, okay, we are going to see two different rules, two rules, and then hereby we are going to study one rule. Okay, so that's it. Now we will come back to here itself. First of all, what does the term genetics mean? What does the term genetics? Okay, what does the term genetics mean? Genetics is the subject. Okay, so genetics is the subject. Okay, which deals with the inheritance, okay, which deals with the inheritance as well as, okay, so as well as the variation, okay, as well as the variation of characters from parents to offsprings. Okay, so this is what genetics is the subject that deals with the inheritance. Okay, so and also you have to know this. Genetics is the basis of heredity. Okay, genetics and then gene, okay, chromosomes, all these are what we can say as genetics. Okay, so it is a subject, it is a branch of biology. Okay, it is a subject which deals with the inheritance. Okay, so where genetics by genes only we are able to express our characteristics. How genes are the things that are something that makes us to express our characteristics? That's a very common question you all might have. I will just clear it out. See, there is central dogma process. Central dogma. What is central dogma? Okay. Central dogma is very simple. DNA is there. Sorry, correct only. DNA, it will produce RNA. Okay. Transcription, translation, then all these we will be studying over there. Okay. First of all, replication. Okay. Then transcription, then translation. Okay. So then at last proteins. Okay, in a molecular basis of biology you will be studying, okay, very interesting chapter, this and that is, okay, both are and very easy. Okay, so this is replication. Okay, so replication of DNA. First of all, central dogma process. Here I will be just giving you the one liner. Already I have uploaded videos for molecular basis of biology. Okay, so central dogma is a process. Okay, it is a sequence of process where first DNA will be replicating itself. Okay, there you will be studying about replication fork, you will be studying about ligases, then you will be studying about lots of things. Okay, fragments of DNA we will be studying, all these we will be studying. Okay, so DNA will be replicating itself. So one DNA double strand will become as two DNAs of double strands. Okay, then this will be giving you, okay, then by transcription, by the process called as transcription, okay, by the process called as transcription. Okay, I cannot write here that much clearly, but the spelling is TRANSCPRIPTION. Okay, transcription. DNA will be converted into RNA by the process called as translation. Okay, so RNA will be converted into proteins. Okay, so what? RNA will be converted into proteins. That's it. So what are proteins? Proteins are the hormones. Okay, hormones are made up of what? Proteins, right? So what are hormones? Hormones are the things that are responsible for our looks, okay, for our characters. If you're short tempered, then because of a hormone only you are getting easily triggered, so that, okay, the angry hormone is being released. If you're too sleepy, then dopamine is being released. Okay, if you are waking up very early, then your melatonin is secreting properly. If your temperature is always correct, then hypothalamus is doing its work in a very good way. All these lots of things as we have studied right in our class 11th itself, based on hormones. Okay, so RNA will be converted into proteins by the process called as translation, and these proteins are what? Hormones, and these hormones are those, okay, which are helping us to be ourselves. Okay, the characters that we have. And since proteins are those which have come from DNA, and DNA is the genetics, okay, and that we have got from our parents, that is why some of our characteristics will be similar. Okay, movies and all they'll be showing, right? Okay, for an example, if there is a daughter and if there is a mother, then the daughter's and the mother's characteristic will be very similar. In movies you will be seeing, or never-ending series at all is there in India, right? Known as serials. There at all they will be showing. So that's what. Okay, they will be actually. Okay, so that's the basic explanation. Okay, and here it is wrong. Okay. Now we will come back to here even. I have just now completed this genetics. Okay, so we will here we have to continue now. So as well as the variation of characters, it is not only just inheritance. See, principles of inheritance and variation is there, right? Okay, so this inheritance and variation is none other than genetics. Okay, genetic deals with both inheritance and variation. That is what they are saying, as well as the variation of characters. Okay, that is why genetics is actually, okay, see, that is why basics of heredity is genetics. Basis of heredity is genetics, because genetics deals with both, okay, genetics deals with both inheritance and variation. Okay, now we will come to heredity. What is heredity? In class 10th we have studied heredity. Very interesting lesson. That is, see, passing of genetic characters from parents to offsprings. Now it is just the passing of characters from parents to offsprings. Okay, now you might have a doubt, that is, what is the difference between heredity, okay, and inheritance? Because the definition is similar, right? But the thing is, here it is a process by which transformation of genetics or characters from parents to offsprings. But here it is not the process. Here it is just the passing. Okay, here you are studying how it is being passed. Okay, and you are here studying what happens. Okay, what is the consequences that will be there, or what are the terminologies, or here you are studying how it is being passed, and here you are studying what will happen if it is being passed. That's it. Okay, then gene. Okay, so what is gene? Actually, first gene was actually called as factor. Gene was actually called as factor because Mendel, okay, Gregor Mendel, he came to a conclusion that something is being passed from parents to offsprings, okay, and that from that generation to the next generation, from that generation to the next generation, from that generation to the next generation, something is being passed. But he was unsure what was that thing, so he termed it as factor. Okay, some factor is the thing that is responsible for all this inheritance and this variation. That is what the thing that is being passed from generation to generation. Some factor is being passed, but he didn't know what was that factor. Later, scientists gave name to that factor, which is known as gene. Okay, now we call it as gene. Exactly same story is what happening to X-ray also. X-ray, okay, a scientist, okay, I forgot that whole story, but I know this alone. That is, a scientist is there, and some rays were causing something, and he called that rays as X-ray. Okay, some sort of X-ray was causing something. Okay, so that is why it is known as like that, but still now no one gave name to X-rays. It is X-ray itself. Okay, next is responsible for our so what is gene? Gene is actually none other than our genetic material, which is DNA, which is responsible for our characteristics, for our traits, for our looks, everything. Gene is actually responsible. Okay, so for an example, eye color, our height, okay, then all these, for all of this, our skin color, our height, all of this, the responsible factor is going to be our gene only. Okay, so responsible for our characters and appearance and inheritance. Okay, for inheritance also gene is the thing that is very important. And one more thing, you just see here in TT, okay, here how many genes are there? Here only one gene is there, here also only one gene is there, here also only one gene is there. Because gene is something that is responsible for a character. Okay, so it doesn't care whether it is short or small. Okay, even small relates with height, tall also relates with height. So here, okay, so here also it has only one gene, here also it has only one gene, here also it has only one gene. Okay, but when you take alleles, okay, alleles are another form of gene. Okay, I think I have written here. Have I? See, variant of gene. Yeah. So allele is actually none other than variant of gene. Okay, where you are having here two different types of alleles. Sorry, two same type of allele. Okay, here also two same type of allele. Here you are having two different types of allele. Okay, here you are having two different types of allele. Here also you are having two, but in these two cases, okay, you are having same type of allele, but here alone you are not having same type of allele. Okay, so that is what it means. Now you come to here itself. Okay, so by the way, allele, it was a term that is said by Batson. Okay, Batson. Now you just come here, that is terminologies. We have heterozygous, homozygous. Okay, go over there, I have written. Okay, so first, what is homozygous? Homozygous is actually a zygote. Okay, zygote is not only for humans, also for plants. Okay, you can say it as embryo, anything like that you can call it as. Okay, so a zygote is formed by the fusion of two gametes which are having identical factors. That means identical genes. Okay, genes. Okay, identical genes. Now then automatically you have to understand that same type of allele. Same type of allele. Okay, now variation. Then one more thing I have to say, that is this concept. Okay, this concept, our Indians. Okay, we came to know it like around the year of. Okay, Indians. We Indians. We Indians had the basic idea about variation. Okay, we all know that variation okay, so we all know that variation is actually caused by sexual reproduction. Yeah, variation is caused by sexual reproduction. Way back. Okay, like why is this not writing? Okay, way back, like in the year of 1000 BC itself. Okay, 1000 BC to 8000 BC. Okay, 1000 BC to 8000 BC. Now, coming to heterozygous. What is heterozygous? It is also a zygote that is being formed by the fusion of two gametes with different. Okay, we're having same type I have written. Having different type of factors. That is what heterozygous means. Now, coming to hemizygous. Okay, it is like hemi means what? Hemisphere we all have studied, right? Hemi means half. Okay, so this is actually having hemizygous means it will have only one gene or half a gene. Okay, how? Okay, why? You take XX chromosome and then you also take XY chromosome. Here Y is also an allele. Okay, Y is also an allele, and here this is also an allele, but these two are completely different. X is different and Y is different. Y is the factor that you can say that is primarily determining maleness. Yes. Okay, so here we are having heterozygous and then hemizygous that I have completed. Okay, hemizygous still, okay, see, I have to explain this still further. So, one thing I have to say, that is chromosomes, we say generally this is what chromosome, right? Okay, but okay, so now we are having another chromosome. Okay, you are having also another chromosome. Now, we all have studied in the cell division itself that this half, okay, and in this half will combine. This half can, that means half of the chromosome will be combining, isn't it? Okay. So when they combine, okay, when they combine, then they will be like this. Okay, so they will be like this. Now when, okay, now when you take in the case of homozygous, that is here also, okay, now you take genes, okay, that is in case of homozygous. If here there is B now, then here also there will be B in the same locus, okay. Locus is singular, so loci is plural, okay. So in the same loci of two different, okay, of this chromosome, okay, in this chromatid if you are checking now, here also it should be B, here also it should be B, then it is homozygous. Here if in case there is capital B, okay, then you can see that now here it is heterozygous, and B can be anything, okay. So why to have B? I will take T itself, very common example. Okay, so here if it is like this or if it is like this, okay, then it is homozygous, or if it is like this, then it is heterozygous, as you can see, okay. But there are a few conditions where here there will be T and here you cannot find T at all, okay. Here you cannot find T at all, but in plants it's not like that. In humans you can find like that, okay. That is X and Y, that is X and Y, okay. In X and Y chromosome you can find like that. Why and how? Okay, you can see, okay, this is what X chromosome, okay, and this is what will be your Y chromosome. Just by the size itself, okay, just by the size itself, we can very easily see it. For an example, okay, for an example, if there is something, okay, if there is a gene or if there is an allele, okay, you can keep it as X allele, then that allele will not be here, okay, will not be here, okay, it will not be here. You cannot find that allele. Okay, so because only one gene is there, okay, only one gene is there, so that means only half a gene. Okay, see, when you are having two of them, okay, when you are having two of them, then only you can say that only one gene. If you are having just one, see, here if there is a gene, then here also that gene should be there, then only we will be saying it as one gene. If in case here is only gene is present and here there is no gene, then it is known as half a gene, only half a gene is there. Okay, that is what XY. And some important thing is, this is an exceptional condition, that is PAR region, okay, PAR region, that is it is a region in where in this chromatid and in this chromatid you can see that region is actually known as pseudo autosomal region, okay, pseudo autosomal region, which is known as PAR region, where in this region alone you can find a few, okay, not a lot, a few genes that are matching, okay. See, here this X is having around 900 gene, okay, around 900 genes, and then this Y will be having around 55 genes, and amongst them not that much, okay, 20 genes, okay, 20 genes are same, okay, 20 genes are same in this PAR region, which is pseudo autosomal region. Okay, now you just decode this word. Pseudo means fake, okay, autosomal cells means body cells, okay. So autosomal means what? It is body cells. So fake body cells in that region, okay. Since, see, generally it is what? It is a sex chromosome, okay, it is a sex chromosome, isn't it? And in this they are having a region where both the genes are same. So that means they cannot give it a name that is based on sex basis, because it is a sex chromosome, and in that sex chromosome there is an exception. So that is why they have given it based on the body cells, okay, based on the body chromosome. That's it, okay. Hemizygous means this. Now, what is phenocopy? One second, I'll just erase it. What is phenocopy? See, phenocopy is actually, they will be having same genotype, okay, they will be having same genotype, but the phenotype alone will be different. That means you take Himalayan rabbits, okay, Himalayan rabbits, where one rabbit in one place. Okay, that means just see, there will be two different organisms, and those organisms will have exactly same genotype. Okay, same genotype. That means same chromosomes they will be having. But the main thing is they will be having different phenotype. Okay, why is it? Actually, it is none other than adaptation. Adaptation. See, you have to change with respect to time. You have to change with respect to time. As even take our case, NEET exam is becoming tougher and tougher and tougher each and every year. So we will have to also improve our knowledge with respect to the year. Okay, like that. Also, these organisms must. Okay, these organisms also should evolve themselves or should adapt themselves to the environmental conditions so that they can survive. It is all just survivability. Okay, fitness test. Okay, next is this we will come to later, but I will just say, since we all have studied most of this in our class 10th itself, it's more likely to be a recap. Okay, so hybrid vigor. What is hybrid vigor? Hybrid vigor is actually superiority over the parents. You just take here, okay, this, okay, capital T, and then this one you take. Okay, see, when you mix this one and then this one, you will get this one, right? Okay, we all know that superiority intends dominant over the parents. So this is also tall. Okay, this is tall, but this is short. You can see that this offspring is actually superior over this parent. Okay, if in case it happens to humans now, then definitely parents will be feeling happier, because parents always wants the best for a child. Okay, so this is what hybrid vigor. Now coming to the next case, these are some important persons. Okay, see, already we have seen this person, now we will come here. Father of genetics. Who's father of genetics? Very important person. Okay, that is our Mendel, Gregor Mendel. Okay, he's having a history itself. Next, father of modern genetics. Here he is just a father of genetics, because see, he is the one and only scientist who went to the ground in sense, he went to the field and he plowed seeds, and then he grained seeds, he gained all those seeds, then again he plowed them, then again he grown them, then he got them, then he resulted. Other than him, most of the scientists will be sitting in a lab with AC, then not in that time there is not AC, now I'm talking about, okay, there will be AC, there will be microscope, they will be watching something, and then they will be doing something. He is the one and only hard-working scientist, and also he has degree in both biology and mathematics. That is why he was able to combine statistics, and logical mathematics. Mathematics with biology. Okay, with biology he was able to combine because he has both knowledge. Okay, next is father of modern genetics is our Batson. He is the person who termed our allele. Okay, and he is the person who said about factors and all those Mendelian laws. Okay, everything he did. Next is father of experimental genetics. Okay, father of experimental genetics is going to be our Morgan. Okay, because he did lots of experiments. Okay, he's like our Mendel only. He's also like Mendel only. But whereas our Mendel is a hard-working fellow and he's a smart-working fellow, because he did that on the fruit flies. Okay, you just take a banana. Okay, then you keep it in your home. Okay, then come after 3 to 5 days, then there will be a family of fruit flies. Okay, they would have there will be a kingdom of fruit flies itself. Okay, grandfather, father, child, great grandson, like that. It will be just going on. Okay, and the scientific name is Drosophila. Very important thing that we are going to study in our future and in the upcoming videos. We have that one big table column itself. Next is father of human genetics and biochemicals. Okay, so is our Gorad. Okay, so Gorad. Okay, and he did research on Alcopturinia. Okay, some sort of name it is. That is Alcopturinia. Okay, Alcopturinia. Now we will come to our Gregor Mendel itself. Our Gregor Mendel is actually, he had born on this age and he died on this, not age, he had born on this lifetime and then he had died in this thing. Okay, and here he did research in 1856 and then he completed his research in 1863. So 7 years of research. 7 years he worked as much as possible. Okay, so his age was actually 34 and then 41. 34 and then 41. Okay, my age only. So 1865. In the year of 1865, actually he experimented in the plants hybridization. So in the year of 1865 is from 20th century. 7 years. Okay, see, first of all, he researched. How did he do his research? He actually used some research. I will be explaining all this, but before that I will say this. He did hybridization. Okay, see, you can see that he did hybridization. That is one thing that we have studied in sexual reproduction and flowering plants itself. We have studied how to do hybridization. Okay, so he did that. Then emasculation, bagging, everything we studied, right? Then he did self-pollination. Okay, pollination. These are the ways how he produced all these three laws. Okay, and law of dominance, then law of segregation is actually those two laws that I said over here. Okay, I said we will be studying two laws. That is law of dominance and then law of segregation. Here we will be studying about law of independent assortment. Okay, then actually he did self-pollination in order to get true breeding. Yeah, correct. True breeding only. True breeding plants. True breeding plants is actually none other than these plants. Okay, these plants are the offsprings of the parents who have done cross pollination with their own brothers and sisters. That means there you take a pot. Okay, a pot of peas. There will be plants. There will be seeds, right? Okay, those seeds. Again you have to plant, then they will be producing flowers, then those two flowers you have to take, then you have to fuse them, then again they will be making some seed, that you have to take. That is what, brother and sisters. And the same thing is what also happening in our fig. Even in our fig also same thing only, that fig was this there, right? That brother will be going and fertilizing his own sisters. How is this? Well, yeah, that is kind of disgusting fact. But because of that, and also because of that, they are also having a problem that is known as inbreeding depression. They develop a problem called as inbreeding depression. And to prevent this inbreeding depression only, lots of plants have evolved lots of techniques. This also I said in the sexual reproduction of plants. So like they will not fuse with their own gametes. That means if a flower is there, if there is a bisexual flower, then that bisexual flower has evolved a few techniques so that that flower will not fertilize itself. If it fertilizes itself then there will be inbreeding depression. Inbreeding depression is none other than some side backs, some side effects. Even in our humans, we are getting lots of inbreeding depression if there is contagious marriage. What is contagious marriage? Marriage between with the same family. That means in Tamil Nadu, in the southern states, it's very common. That is, if your grandparent, for an example, there is a grandparent. That grandparent has a boy child and a female child. Then that boy child will again have some kids. That female child will again have some kids. Right? Now that male, that kid male will actually should marry this kid female. That is what the contagious marriage is. Within the same family, they will be making marriage. Because of that, lots of problems, side effects. Like 6 fingers or sometimes short finger. Then some mental issues, chromosomal issues. Then long-term cancers. Cancer is actually so long-term diseases like that. Lots of lots of side effects are there. So this is what he did. And also, in his age, he actually published a book. He published a book, but in that time he wasn't able to get success. He wasn't able to get success. So because of that, his book was forced. He cannot sell his book that much. And also he was able to sell it only to his locality, and he thought, okay, he published the book and he thought, okay, that's it, his job has completed. And in his locality there wasn't that much literates, and whoever read it, even those literates, thought, what nonsense is it? No, what nonsense is it? Okay, it is actually having mathematics and biology, and in that time the combination of mathematics and biology is not that much wide. Okay, it wasn't that much, so that they just thought, okay, he's some sort of idiot, he just did something, so they just neglected it. Then after that, a few scientists came, okay, in the future, then they researched on his laws, and then they said it is all correct. Then only he got his name. Okay. Now whatever I've said, like all those things, right, that scientists and that year, okay, in the 20th century only was accepted, then okay, and scientists, and in the future videos there will be, okay, in the future lectures there will be, I will say those. Okay, then also he did self-pollination so that he can get true breeding. Then after that he did cross-pollination. Okay, so he had self-pollination, cross-pollination, then also artificial. Actually, all these two are artificial pollinations. Okay, what are these? He did artificial pollinations, which are actually of two different types. That is self-pollination and then cross-pollination. Okay, experiment in plants hybridization. Yeah, he did experiments in plants hybridization. That is what all these are. Okay, so that's it for here. Next is, these persons did experiments in these things, is like Carl Coins, then Hugo de Vries, then wait. Flowering plants, H1 Texmark. What names are these? Very confusing, right? But we have to remember all these. Okay, see here, true breeding lines, and he got all those true breeding lines by emasculation and then bagging. Okay, emasculation is just cutting off stamens. Okay, bagging is the covering of stigma. Okay, it's just covering of stigma on the female reproductive part of the flower. Okay, you come here itself. That is, Carl Currens is actually nice researcher. Okay, then evening primrose, flowering plants. Okay, so Carl Currens is a person actually in the year of 1864 to 9100. Sorry, 1894 to 1900. He did some researches on maize plants based on some principal rules, like zinnia, like that there are some principal rules. Okay, and then he used Mendel's principle. And also same, Hugo de Vries was also a person who researched on evening primrose. Is a very important thing, you know. It is actually, see, our Mendel said that there is no blending of characters. That means two different characters will not blend, but that is not correct. Okay, that is not correct. Okay, he didn't inform anything about blending of characters. But primrose, okay, but evening primrose is not like that. Okay, it is a flower, okay, which has blending of characters. That means, see, for an example, you take pea plant, okay, you will get T and T, right? Okay, but it will not happen. It will not happen here. For Hugo de Vries, it's like, okay, see, in evening primrose, when you take, see, when you take a pea plant, okay, in the tall plant you are having and the small plant you are having, and when you combine you will be getting a tall plant. But when you take a primrose, okay, in that you take a white rose and a red rose, then you will be getting a pink rose. How is it? Very good, right? Okay, then next is exactly, see, for flowering plants, or most of the flowering plants he did research. Who is our some person like that, that is Irish one Tishmak 6, okay, a person like that. See, most of the people are Chinese or Koreans. Okay, now next is, we will talk again about Mendelian characters. Okay, now see, he has totally taken 14 plants. Okay, 14 plants. All 14 is actually with different characters. See, one plant is actually tall, then another plant is actually short. One plant is actually having flower position as axial. What is axial position? When you take a flower, okay, when you take a flower, okay, in the stem, if the flower is coming here, then that is axial. Or if that flower is coming here, then it is known as terminal. okay, in the terminal stem. Then flower color, okay, if it can be either violet or yellow. Then pod shape, okay, the pod of the shape of the pod can be either full, that means like it need not be constricted, it will be blotted, okay, the pod will be blotted, or it can be constricted. Then okay, the pod color can be green and yellow, okay, and when you check this, okay, this and this will be completely opposite. Both of this will be actually completely opposite, the colors I'm talking about. Okay, then also sheet shape, either it can be round or it can be wrinkled. Wrinkled in sense will be like this, not round shape. Okay, so that is what it is. And also these characters are in some specific chromosomes, okay, are in some specific chromosomes, like this, okay, this character is in 4th chromosome, this character is in 4th chromosome, this character is in 1st chromosome, this character is in 4th chromosome, this character is in 5th chromosome, then this character is in 7th chromosome, okay, and then this character is in 1st chromosome. Okay, like all these characters will be located in the loci evenly of a chromosome. Then now we are having Punnett square, okay, it is also known as checkerboard. It was actually designed by Reginald C. Checker, okay, Reginald C. Checker, okay, where in the vertical row, that means if this is a Punnett square, then in here you have to put male, here you have to put female, okay, then you will have to cross them. Then that's it. This is what Punnett square mean, okay. Then oops. See those 3 rules, okay, those 3 rules that is given by Mendel, okay, Mendel has given a few rules, right, where are they? These 3 rules are the basic rules of the entire inheritance itself, okay. Based on these rules, actually in the initial time period it wasn't accepted, but later on it was accepted by further research of some other people, okay. Then one common question that they can ask is, one very common question that they can ask is, why did Mendel choose, okay, why did Mendel choose Pisum sativum? What is Pisum sativum? Pisum sativum is our pea plant. Okay, is our pea plant. Now, there are a few things that is, first of all, pea plant is actually having a short lifespan. And they can easily produce offsprings. See, first of all, a plant, okay, a desirable plant, okay, or an ideal plant for this condition, for his research, should be having less lifetime, because see, if a plant is taking about 1 year to produce offspring, then when will he complete his research? So that is why he has taken a plant that is actually able to produce offspring very soon, okay, as soon as possible. That is the reason, okay. And also maintenance should be very easy. Is easy. And also that plant shouldn't be very large, okay, that plant shouldn't be very large, it should be a small plant. Then also characters, okay, its characters are completely contrasting, okay, there is no blending of characters, okay, so it has, okay, contrasting characters. Okay, contrasting characters that is easily visible, like you need not to have a microscope to see them, just with your naked eye you can see it, okay. Then these are the things that made them, okay. So inheritance of one gene, actually what he did is he cross-pollinated a tall plant. And then a short plant. Okay, then when he cross-pollinated for the first time, he got a tall plant. Okay, then again when he cross-pollinated, and he was shocked, where is the character of short plants? Okay, he was shocked. Then again when he cross-pollinated this with again the same brother or sister. Okay, this time he got tall plant. Okay, and then short plant. Okay. See, in that time he didn't know what are these factors. He didn't know what is this small tt. Okay, and then he didn't know what is this. Okay, but he came to a conclusion. He's a very brilliant fellow. So he came to a conclusion that is, okay, here one character is suppressing another character. Okay, he came to know that one character is actually suppressing another character, and the suppressed character, okay, and then the suppressed character the suppressed character, then the dominant character or the unsuppressed character I can take, right? Array. And the unsuppressed character, okay, in this, okay, he named the suppressed character as recessive character. Then this unsuppressed character was named as, okay, this unsuppressed character was named as dominant character. Okay, and also when he was doing this research now, in that time, this tall plant was actually, if in case there were 4 plants, now amongst that 3 plants were tall and 1 plant was short. Okay, so this is what said as phenotypic ratio. Always this was actually constant. Okay, no matter how many plants were there, amongst them 3 parts were tall plants and 1 plant was actually, 1 part was actually short plant. Okay, so this is actually, and also he noted one more important thing. Okay, that is generally what will be human mindset, what will be a human thinking, that is when you are taking a tall plant and when you are taking a small plant and when you are mixing them, there will be a plant with moderate height. It will neither be tall, it will nor be short, right? But here it is an exception. So he came to know that there is no blending of the characters. But you might ask me that there are a few plants which has this blending character, right? That means a few plants are actually for example, that primrose, okay, that evening primrose is there, right? It is actually getting pink colored flower. And also when you take a mango, a big mango, and when you're taking a small tasty mango, and when you're taking a big untasted mango, and when you're fusing them, then you'll be getting a moderate sized good taste mango, right? Okay, so that one over there also blending is what happening. So then how is that alone happening, Kannan? Well, actually the main reason is over here there is no blending of characters because, okay, dominant, okay, because of the dominant character, okay, tall dominant character, where it is highly dominant. Here this character is highly dominant, that it does not allow other character to express themselves, okay. So there will be a lot of people even in the class that they will be very dominant and they will not allow others to be at least a bit dominant, okay. So they will be suppressing others, like that there will be a few people in the classroom, okay. So no blending of the characters, and here it is because this is actually, this tall is highly dominant. Because of that, this short character is not visible. So, and also there is actually co-dominance. Okay, this I will be saying in a very detailed way itself in the upcoming videos. We will just see the co-dominance. Okay, then what is one more thing? The incomplete dominance. I'll just give you one good example. Co-dominance, now 50% dominant, which means, okay, you just take a black cow, then you take a white cow. Now you just fuse them, then you will see. If in case this cow is having a co-dominant gene, then you will be getting a brown colored cow. Okay, then if in case it is having an incomplete dominance, I don't know what color, okay, whether it is incomplete or whether it is complete, then you will be getting black and white cow. Okay, just like the cow that Krishna has, na. So in movies at all, if in case that cartoon, okay, then or in movies, even in pictures at all, Krishna will be having a cow beside him, and that cow will be blackish and then whitish. Okay, some spots will be white, some spots will be black. Like that, okay, you will be getting. That is the difference. And here you can see that even though black is more dominant than brown, sorry, black is more dominant than white, but here it is not 100% dominant. Here it is just 50% dominant. That is what happening here. Okay, that is what here it is happening. Here in some places black is highly dominant and in some places white is dominant. Okay, here it is incompletely dominant. Okay, then over here our tall is actually what 100% dominant. Okay, I think, I don't know whether is this there or not, but when people are studying higher studies now based on genetics, there I think there is a degree of calculation. There is a degree of calculation for this dominance. Okay, dominance. If that dominance degree is very high now, if the dominance degree is very high, then in that time it will be completely dominant. Okay, just like a tall and small. Okay, just like the tall and small. Like the smallness, the recessive character will not at all be expressed. Okay, I think it will be there in the highest degrees. Based on that degree only, we will be determining whether it is tall or short. Okay. Okay. Then Mendel, we have seen Mendel. Then and also there are some rules. Okay, that is always this must be in small letters. Small letters. Your recessive characters must be in small letters. Then this unsuppressed character or this dominant character should be in capital letters. Okay, should be in capital letters. And then, okay, so then after this, now coming to the genotypic and then for monohybrid. Okay. By the way, wait, I will just give you a good monohybrid cross. what is monohybrid cross? Okay, that's what is monohybrid plant. Okay, what is monohybrid plant? I will say you are having this, okay, and then now you have got this, okay, then again you are fusing this with another plant, then you are getting okay, now you are getting like this. Now this is one known as monohybrid cross because you are just having, see, this is what hybrid, this is what hybrid is. This is pure, but this is hybrid. Okay, so yeah, okay, good. This is what monohybrid, that's it. Monohybrid, this is just having only one gene. Okay, whatever is having only one gene, then that is known as monohybrid. If in case something is having two genes, now then we will be calling it as dihybrid. Okay, it will have only one gene. So that is what we will be calling as monohybrid. And here the phenotype and then the genotype. Then the genotypic ratio. Okay, the phenotypic ratio and then the genotypic ratio. So the phenotypic ratio is actually based on the physical appearance. Physical appearance. Okay, genotypic ratio is actually based on the genetic genes. So see, we will just do it. Physical appearance in sense, okay, it is tall is to tall plants is to short plants. So how many tall plants are there? All these are tall plants, isn't it? So out of 4 plants, 3 are always tall and then 1 is actually small. This is what your phenotypic ratio. What is your genotypic ratio? Genotypic ratio is actually based on the genes, that is, okay, purely tall, is to hybrid tall, then short. Okay, so this is what purely tall, that is just only one gene is there and then 2 alleles of same thing, that is homozygous. Okay, it should be homozygously tall also you can write, that is, okay, only one is there. Then hybrid tall is 2 is there, okay, that is heterozygously tall, that is 2. Then again, okay, tall, that is just short. Okay, there is no pure short or hybrid short, that is just this. Okay, so 1 is to 2 is to 1, that's what it is. So you just take this, okay, that is only 1 T is there in among and also along with this, okay, 2, this is there, okay, then also along with this only 1, this is there, isn't it? Okay, isn't it? This is correct, right? This is what it means. Okay. So how is this? Isn't this looking like this? we will just, okay, elaborate it. Which color? This color. The color is TT. Okay, just first T square will be there now. Okay, T square, then this square, then also along with that, what is there? Okay, then 2 into T into this. Okay, so how can I write this? I can write this as like this. Okay, yes, right, I can write it like this. So it is just a binomial expression. Okay, so this is a binomial expression. Now, types of test. With this, we will complete this video. Okay, so what is this types of test? See, types of test. See, first I will say why is there. No matter how much a person can be perfect, but when a person is researching with all these types of plants, okay, he will be just combining all these will be happening, and then in between, definitely he will be getting confused with what type of genotype is there. Okay, what type of genotype is there? Whether it's this. Okay, so for that, what people use is actually test. They just test it, and then by using that test, they just find whether is it TT, capital TT, or capital T and small t, or is it just small tt. Okay, so you are having a few types of test. Okay, so back cross is there, out cross is there, then test cross is there. Okay, so you are having out cross, then you are having back cross, then you are having out cross. Now we will just check what are these. Okay, my leg started to pain from morning. This is my second seminar, so I don't know how much time am I taking this seminar, but 2 hours of continuously I'm standing, and I'm proud of it. Okay, so first back cross is none other than F1, okay, into, wait, I will just say what is F1. See, this one, okay, this one generation is actually, this is what parental generation, we say it as parental generation. Then this one is known as F1 generation, this one is known as F2 generation, where F means filial generation. Okay, filial generation. So that is what it is. So F1 into any parent, you have to do it as F1 into any parent. This is what known as back cross. Okay, this is what back cross. Now for out cross, then what it is? Okay, for out cross it is F1 into dominant parent. Here completely opposite. It is like F1 into recessive parents. Okay, by the way, I will just write it. Recessive parent, dominant parent, then F1 generation. Okay, so what is F1 generation? F1 is actually none other than capital T small t. Okay, recessive parent is actually this one, then this one is this one. Okay, now we will just cross it. So F1 into dominant parent. What is dominant? First, what is F1? capital T into small t. Dominant parent is this one. So this is actually okay. So when you cross pollinate, what you will be getting? You will be getting this is what you will be getting. Okay, everything will be tall. All tall. Okay, so all tall. Well, actually, F1 is what your unknown. Okay, F1 is what your unknown. F1 is what your unknown is. And also, okay, like moreover, how can I say? See, F1 is unknown. You don't know what it is. Okay, it can be also small tt. There is no problem with that. It can be also small tt or it can be just capital TT. Well, dominant parent and this is actually constant. You have to find the F1 only. F1 need not to be always capital T and small t. Okay, see, you don't take this exact thing. Okay, this one is the dominant parent is fixed, recessive parent is fixed, but this F1 is not fixed. Okay, but this F1 is not fixed because there is no such condition that you have to cross pollinate this and this. You can also cross pollinate capital T, like homozygous tall and homozygous tall, or homozygous short, homozygous short, or homozygous tall and homozygous short, or you can also cross pollinate homozygous tall. Okay, like this you can cross pollinate anything. Okay, it need not to be like only this one and this one. Okay, so this is why this is your unknown. This is what you don't know. Okay, if this unknown is capital T small t, then you will be getting everything as tall. This is case 1. Okay, and if this unknown is actually like this you keep, then also everything will be tall. And if this unknown is, then also all tall. See, so no matter what, okay, when you cross pollinate, okay, without cross, then everything will be tall. So okay, then everything will be tall. Now coming to, okay, back cross, that means it is just you are crossing again with the parents. Okay, F1 into parents. F1 can be anything, that we don't care. Now coming to out cross. Okay, so what is your out cross? Out cross is actually, wait a second, into dominant parent. What is that out cross? I written again out cross. It should be test cross. It should be test cross. Okay, so what is a test cross? Test cross is actually F1 into recessive parents. So recessive parent is just this. Okay, into F1. So F1 is what your unknown is. So for an example, I will take F1 to be this one. Okay, then you will be getting it as okay, like this you will be getting. Then if your F1 is like this, see, you will not have confusion with short plant, because short plant, and you know it is always small t. Okay, so you will not cross pollinate it with short plant. Okay, you will have confusions only whether it is homozygous tall or heterozygous tall. Okay, that only you have to decide. Okay, so here this is what will be used. Test cross is what you used to determine whether it is homozygous tall or or heterozygous tall. Okay, then it can be like this. Now you just see. Okay, see, when you are cross pollinating like this, okay, and when it is heterozygously tall, then half of the thing, okay, then it will be 1:1. It will be 1:1. Then when it is 1:1, okay, half of the plant will be tall, okay, half of the plant will be tall and half of the plant will be short. Okay, when it is like this, then you can determine that it is heterozygously tall. okay, but when everything is tall, then you can determine that F1 is homozygously tall. That's it, okay. And outcross is actually like you can do this and this, then you can do, okay. That is not a thing, okay. You can take like this is a outcross is a type of back cross, test cross is also a type of back cross, like that you can keep it as, okay. So that's it for today's video. We will be meeting in another amazing video. Till then, bye bye.